Hello everyone. Today we will discuss about pyruvate dehydrogenase complex. It is a multi-enzyme complex. Multi-enzyme complex, and it catalyzes the conversion of pyruvate to acetyl CoA. Pyruvate to acetyl CoA. The overall reaction involves both oxidation and reduction. So, it is named as oxidative decarboxylation. Oxidative decarboxylation. And this reaction is catalyzed by a multi-enzyme complex that is known as pyruvate dehydrogenase complex or PDH complex. This pyruvate, which is formed in cytosol, and it is transported to mitochondria. This is by a transport protein. This pyruvate dehydrogenase complex, it is a connecting link between glycolysis and TCA cycle. It is a connecting link between glycolysis and TCA cycle. This enzyme complex consists of 3 enzymes and 5 co-enzymes. It consists of 3 enzymes and 5 co-enzymes. So, first we will see the 3 enzymes. Enzymes are pyruvate dehydrogenase, pyruvate dehydrogenase. Second enzyme is dihydrolipoyl transacetylase. Dihydrolipoyl transacetylase. That is the second enzyme. Third enzyme is dihydrolipoyl dehydrogenase. Dihydrolipoyl dehydrogenase. So, these are the 3 enzymes in pyruvate dehydrogenase complex, and this complex also contains 5 coenzymes, and these coenzymes are, first one, five coenzymes. First one is thiamine pyrophosphate or TPP. Second one is lipoic acid. Lipoic acid. Third one is CoA-SH, then FAD and NAD+. These are the 5 coenzymes of this pyruvate dehydrogenase complex. So, let us see the steps. In the first step, pyruvate is decarboxylated to form a hydroxyethyl derivative of the thiazole ring of enzyme bound TPP. Here, pyruvate, it is decarboxylated and it forms a hydroxyethyl derivative. Decarboxylation means removal of carbon dioxide, and with TPP, that is thiamine pyrophosphate, it forms hydroxyethyl TPP. Hydroxyethyl TPP. And here comes the first enzyme, that is pyruvate dehydrogenase. Enzyme is pyruvate dehydrogenase. That is the first enzyme in the pyruvate dehydrogenase complex. Pyruvate dehydrogenase. This hydroxyethyl TPP, it is known as active acetaldehyde. It is otherwise known as active acetaldehyde. So, this is the first step in the conversion of pyruvate to acetyl CoA. This is active acetaldehyde, this hydroxyethyl TPP. In the next step, this hydroxyethyl TPP reacts with oxidized lipoic acid and it forms acetyl lipomide or acetyl lipoic acid. So, hydroxyethyl TPP then reacts with lipoic acid. It reacts with the lipoic acid. Oxidized lipoic acid. This is the oxidized form, and it forms acetyl lipomide or acetyl lipoic acid. Acetyl lipoic acid or acetyl lipomide. Then in the presence of dihydrolipoyl transacetylase, that is the second enzyme, this acetyl lipoic acid then reacts with the CoASH. It reacts with the CoASH and it is converted to lipoic acid, that is reduced lipomide, or that is known as dihydrolipoic acid. Dihydrolipoic acid, and this CoASH, which is converted to acetyl CoA. This acetyl lipoic acid, acetyl CoA will become acetyl CoA. That is the formation of acetyl CoA from pyruvate. So here comes the enzyme, the second enzyme, that is known as dihydrolipoyl transacetylase. That is the second enzyme. So that is a step. This acetyl lipoic acid in the presence of enzyme dihydrolipoyl transacetylase, this acetyl lipomide or acetyl lipoic acid reacts with the CoASH and it forms acetyl CoA and will form reduced lipoic acid, that is known as dihydrolipoic acid. Then this reduced lipoic acid, that is dihydrolipoic acid, is reoxidized by FAD. It is reoxidized and it forms lipoic acid, oxidized lipoic acid. This is by FAD, and it will form FADH2. So this reduced lipoic acid, that is dihydrolipoic acid or dihydrolipomide, it is reoxidized by FAD by the enzyme, that is a third enzyme, dihydrolipoyl dehydrogenase. Dihydrolipoyl dehydrogenase, and it is reduced back to, it is oxidized to lipoic acid. So here FAD is converted to FADH2. Finally, this reduced FAD, that is FADH2, is oxidized by NAD plus, and it is oxidized by NAD plus, and it forms NADH plus H plus, and it transfers the reducing equivalent to electron transport chain. So finally, the reduced FAD, that is FADH2, is oxidized by NAD plus and it forms NADH plus H plus. We know that the NADH which will enter into ETC and it will form ATP. So from one molecule of glucose, we will get 2 molecules of pyruvate. So from pyruvate, 2 molecules of pyruvate, we will get 5 ATP molecules. 5 ATP molecules. So from 2 molecules of pyruvate, 2 molecules of NADH. From 1 molecule of NADH, it is 2.5 ATP, and from 2 molecules, it is 5 ATP molecules. That is the energetics. So that is about the pyruvate dehydrogenase complex. So coming to the regulation, of this pyruvate dehydrogenase complex, this PDH is active in dephosphorylated state. PDH is active in dephosphorylated state and it is inactive in phosphorylated state. This PDH, it is active in dephosphorylated. And it is inactive in phosphorylated state. Phosphorylated. Then ATP, which causes phosphorylation with the enzyme PDH kinase. So, phosphorylation. ATP causes phosphorylation of this PDH active to inactive form. So, it will form inactive. It will become inactive phosphorylated form. ATP, ADP with the enzyme PDH kinase. PDH kinase. So, it will become inactive. Then products like acetyl CoA and NADH. Acetyl CoA and NADH. These are the products of this pyruvate dehydrogenase complex. They inhibit PDH complex. That is known as regulation by products. Regulation by products. So, products like acetyl CoA and NADH, this inhibits the PDH complex and it is known as allosteric regulation by products. Then this another enzyme known as PDH phosphatase. With this PDH phosphatase, it converts inactive to active form. This is by dephosphorylation. That is, the enzyme is PDH phosphatase, and this will become active. Then insulin. Insulin stimulates this phosphatase enzyme, PDH phosphatase enzyme, and this PDH phosphatase enzyme which converts inactive to active form. So, insulin stimulates, or insulin helps in the formation of active form. Then this PDH kinase enzyme is activated by rise in ATP ADP ratio, ATP to ADP, then NADH to NAD plus, NADH NAD plus ratio, then acetyl CoA to CoA-SH, acetyl CoA to CoA-SH ratio, then increased cyclic AMP. These all will increase or enhance the activation of PDH kinase. Then this PDH kinase is inhibited by increased pyruvic acid. It is inhibited by increased pyruvic acid, that is by carbohydrate diet. Here it will form active PDH. Then in starvation and diabetes mellitus, this diabetes mellitus, acetyl CoA and NADH, which is produced by beta oxidation. By beta oxidation, it will activate this PDH kinase. So, there will be a decrease in active form. That is in starvation and diabetes mellitus. In starvation and diabetes mellitus, by oxidation, beta oxidation of fatty acid, this acetyl CoA and NADH will be produced, which will activate PDH kinase. So, there will be a decrease in the active form of PDH. Coming to the importance of this PDH complex, it is a committed step in oxidation of glucose. Committed step in the oxidation of glucose. Then it is a connecting link between glycolysis and TCA cycle. Glycolysis and TCA cycle. Then in chronic alcoholics, there will be a deficiency of thymine pyrophosphate, that is TPP, and this will inhibit this PDH complex. This will lead to accumulation of pyruvate and lactate. So, in chronic alcoholics, there will be deficiency of thymine pyrophosphate, which will inhibit PDH complex, and this will lead to accumulation of pyruvate and lactate. This is seen in Wernicke-Korsakoff syndrome. Wernicke-Korsakoff syndrome. So, that is the importance of this PDH complex. So, this is about pyruvate dehydrogenase complex. This is today's topic. Thank you for watching.